All right, hello everyone, and welcome to STAT's virtual event, ASH Recap Live, sponsored by Janssen Oncology. Thank you for joining us. I'm Damian Garde. I'm a reporter here at STAT, and I'm sure you are familiar with how this works by now, but before we get started, I do have a few quick housekeeping items. Hello everyone, and welcome to STAT's virtual event, ASH Recap Live. I'm getting my own audio back to me. There we go. So, housekeeping. At the bottom of your screen, you should see several icons. You can use the chat feature to chat amongst yourselves in the audience or to send a message directly to the STAT team, but please do not put your questions for us there in that chat. However, we very much want your questions, and if you have them, you can use the Q&A feature to type a question, and we, the moderators, will see a list of them, and we will try to answer as many as we can during the event. In that Q&A window, you will also be able to see questions that other attendees have asked, and you can upvote the ones that you would most like to see answered. And we'll be taking questions throughout the chat, so please don't wait until the end until submit them. This is what makes these events engaging and fun, and we want to hear from you, so be on that. Final thing, please note that this will be a two-part event. First, I will be sitting down with Dr. Mikhail Sakaris, and then my colleague Angus Chen will sit down with Dr. Laura Michaels. Before we get started, I would like to welcome Tyrone Brewer, who is president of U.S. Oncology at Janssen Pharmaceutical Companies of Johnson & Johnson, for a sponsored introduction of today's event. Please note that these remarks have been pre-recorded.
It's so important that we keep this discussion about clinical information. Thank you to Stat News for the opportunity to join you today and introduce this session. It's so important that we keep this discussion about clinical information and medical advancement at the forefront and maintain a critical eye. At Janssen, we value the opportunity to remain connected with you and through forums like this as ongoing partners and working through medical challenges. Although we work in our respective institutions, we're all part of the oncology community with the same goal to bring a cure to cancer patients. When we come together in such important forums, such as ASH every year, we have focused time to learn from each other and to experience that, you know what, we are actually making progress for patients. This was my first ASH meeting as a part of Janssen Oncology, and it was inspiring and energizing to be there and to see the amazing science happening to advance care for patients. Every one of us fights so hard to free patients, caregivers, and healthcare professionals of the grip that cancer has on so many of our lives. This is both a global and personal cause. While this unified purpose can be daunting, it's something that we can all be proud of. At Janssen Oncology, our mission is to reimagine care so patients can redefine living. This underpins our work every day. It keeps patients at the center of everything we do and captures our bold aspiration on their behalf. We take our mission very seriously. It's just not words on a page, but it's something we embody. We're always listening and learning from people whose lives are touched by cancer. And in lifting patient voices, we're lifting our ability to serve them, and we believe change what a cancer diagnosis actually means. We're reimagining treatments, collaborations, clinical practice, care delivery, and the way people navigate the cancer journey to help patients live longer and, more importantly, live better. Our commitment also goes beyond medicine. We're working to reimagine how we connect and collaborate with all stakeholders through care delivery and ingenuity by pushing boundaries. If we do this right, then the impact goes back to patients, and we can redefine living. And for them, they can redefine living in their own way and on their own terms. Our mission also inspires us to stay focused on the value of partnerships and organizations and individuals like those of you who are here today. We remain forward-looking so that in working with the cancer community, we can realize our potential. And we need to continue to address healthcare disparities. One notable example of our mission in action is focused on addressing health disparities in multiple myeloma. Multiple myeloma disproportionately impacts Black people more than any other race or ethnic group. Because it's uncommon and because of challenges within the healthcare system, multiple myeloma is often overlooked, especially in Black people. With this knowledge, we started That's My Word campaign, which is focused on creating better health outcomes for those at risk of multiple myeloma within Black communities. As a part of the campaign, we're bringing together Black communities, care leaders, healthcare professionals, and advocacy organizations with the goal to collect 100,000 promises to learn more about multiple myeloma, spread awareness, and encourage those at risk to be proactive with their health. We're working with advocacy groups and role models, namely comedian D.L. Hughley, to provide education and empower self-advocacy through a simple promise at the community level to help us reach and hopefully make a difference in high-risk communities. Bringing this back to our mission, this is just one example of how we're finding new ways to ensure we reach all patients by meeting them where they are, in their communities, and making these connections meaningful. Our goal is not only to support patient outcomes through transformational therapies, but is also to support where the journey begins. The other critical aspect of our mission is that it gives us a framework to set for our future strategies and take the right steps to get there. Namely, we're leading innovation towards an aspiration of cure. We're aiming to develop complementary and combination of a portfolio of immunotherapy treatments that will hope to transform multiple myeloma into a chronic disease. Not all myeloma patients are the same due to the underlying heterogeneity in myeloma. This presents challenges in terms of treatment choices, and having targeted therapy options is critical. For a complex disease such as multiple myeloma, we are intentionally about expanding our portfolio to address unique cellular targets through complementary and combination of immunotherapy regimens. We have an industry-leading multiple myeloma, myeloma pipeline and portfolio with our Janssen products for every line of therapy, from frontline to heavily pretreated relapse refractory patients. So that's where the journey begins and beyond, and we're looking to maximize our impact. Data generation is going to be critical. It helps to keep moving the needle and keep gaining clinical value and a more deeply understanding of the effects of treatments on various populations. At ASH, we were thrilled to present over 50 abstracts across our deep pipeline and portfolio of products. Over the years, we have continued to increase the diversity as well as the scope of data that we present, which this year included company-sponsored studies as well as investigator-initiated studies and real-world evidence. And what we care about most is that this data is of high quality and that it holds meaning, both for every patient with the corresponding disease, as well as to physicians who may have to consider data implications for their practice. It's also important that we're actualizing on our commitments to diversity and health disparities. We focused on representing all demographics and high-risk populations. Our overall presence showed movement towards the future of multiple myeloma care and how we're thinking differently and looking at individuals. This is no longer one size fits all as it used to be. We look at three things coming together for each individual: right patient, right time, right treatment. Revamping treatment and changing the paradigms of care are important and urgent now. And yet, we're really just looking for one day trying to find a cure. That's really the end goal. And that's why we continue to be inspired by our mission to reimagine care so patients can redefine living. Thank you again for this opportunity. We're always thrilled to collaborate with you to foster better care and outcomes for patients today and tomorrow. And thank you for all of your contributions to this community. Our work together helps us to do more for patients, and that's what matters most for all of us. I hope you enjoyed today's event.
All right, thank you, Tyler, for that introduction. And here to join me for part one of today's discussion, please welcome Dr. Mikhail Sakaris. He is Chief of the Division of Hematology at Sylvester Comprehensive Cancer Cancer Center, which is part of the University of Miami Health System, and over at the American Society of Hematology, he is chair of the Communications Committee. Dr. Seckers, thanks for joining us.
Thank you so much for having me, Damien. It's great to be here.
So a lot of the interest in ASH is tied to new interventions, small molecules, antibodies, cell therapies, and that might change the standard of care in hematology. And at this event, we'll get to more of those that were presented at the conference, but I think it'd be valuable to start by talking about the important work of examining whether that standard of care should indeed be standard in the first place. I'll ask you about some specific studies, but I wanted to ask, maybe as an introduction, you've described some of this work as challenging and sometimes dispelling the dogmas of hematology. What does that mean to you?
Yeah. So thank you for that question. I love this area of research within hematology and oncology. I think that with some of the fruits of our successes within HemOnc, we've been able to take a step back and say, whoa, wait a second, maybe we're doing a little bit too much here. Maybe we can back off and people can have exactly the same outcomes. The other aspect of this is that, just like I think in any professional field, there are rumors that are perpetuated and perpetuated and perpetuated, and then they become truth. And when you try to dive into that so-called truth, you find references to previous review articles, to previous review articles, previous review articles, and you find that there's actually no data supporting that practice. So we put together at ASH a session specifically on challenging the status quo, on really doing the formal studies to say, wait a second, are the practices that we've been doing for so many years really the right approach, or should we take a step back and reassess?
Got it. So I wanted to start with I was reaching for the word sensuous. I think that's probably wrong. A very vivid study presented at the conference that stood out to me, and I think to a lot of people, involving not drugs, but food. So tell us about the dogma of diet when it comes to patients undergoing stem cell transplants and what we've learned about it.
Well, you start off with the word sensuous, Damien. You kind of set me up for failure. I don't know that there was much at ASH that was sensuous, but this is, I guess, as close as you can get. So for decades, we have been working under the philosophy that people who have significantly compromised immune systems are obviously more susceptible to infections. Well, that was brought out in spades during the COVID pandemic and the just awful outcomes of people with compromised immune systems when they actually did catch COVID. And one of the ways that we've been intervening on this to try to mitigate that is by limiting the types of foods that people have been exposed to. The theory has been that foods have the potential of containing some bacteria. If a person eats those foods with that bacteria and the person has a compromised immune system, then that person could die potentially of a bacterial infection. That's one aspect of it. The other aspect of it is that patients who are undergoing hematopoietic cell transplantation have a particularly vulnerable immune system because they have a compromise in both myeloid and lymphoid function. So they're really susceptible to just about anything. And that not only would that food expose them to the potential for infections, but also could have an impact on their rates of developing graft versus host disease of the gut, which of course is a terrible morbidity that can follow the transplant itself. So given all of that background, and I will tell you, I have not been a believer in this. So I'm right now at the Sylvester Comprehensive Cancer Center at the University of Miami. Prior to this, for 18 years, I was at Cleveland Clinic in Ohio. And years ago, I did away with the neutropenic diet on our leukemia floor because anecdotally, but with a lot of anecdote, the longer I get, I had never seen a patient die from a bacterial infection from food. So this was a great study because it's so validating and because it's really going to have an impact on our patients' quality of life. In this study that was conducted in Italy, 224 patients were randomized to receive a neutropenic diet, which interestingly, the authors refer to as a protective diet, right? And there's that notion that we're protecting patients from harm. I think there's the additional sensuality, if you will, of the fact that there's emotion around food. And
not a coincidence this study was conducted in Italy, I assume.
That's right. They didn't comment on whether the Italian protective effect of wine was in play also. So, but I will tell you that when I am, walking this journey with patients and their families, one of the most common questions I get is, can I alter my diet to somehow alter the course of this of this disease and thereby altering the course of my own fate? People are really desperate to do something to control this cancer that they've gotten through no fault of their own. And family members in particular are so desperate to try to help and do something. They always ask me, what is it that I can do about diet? How can I cook differently? How can I give my spouse something different that will somehow help him or her? So this is actually a real emotional issue. So 225 patients were randomized to this protective diet versus a non-restrictive diet. It took a few years to enroll all these patients to the study. I wonder if there are a lot of people who weren't willing to go on to the study because they have such an emotional connection to food and didn't want to be randomized one way or the other. The investigators then followed the patients for a number of different outcomes. And the bottom line is they found absolutely no difference in these outcomes, whether patients received this, protective diet, I would call it a restrictive diet, versus a non-restrictive diet. And when I talk about outcomes, they looked specifically at rates of infections, at rates of severe infections, at rates of sepsis, at fever of unknown origin, and at graft versus host disease and survival. None of these was different between those who got the neutropenic diet and those who received the non-restrictive diet. So the bottom line conclusion here is you don't need to restrict people's diets.
Yeah, I found that particularly fascinating. And also I was unaware, I was familiar with the concept of neutropenic diets. I wasn't familiar with just how restrictive they were, not only in terms of the foods you could not eat, but also the, violence you had to do in the kitchen to the foods that you could eat, which is sort of neither here nor there. But so moving from cuisine and into therapeutics, and yet another kind of dogmatic thing, there's been quite a lot of discussion and debate in recent years about who gets to participate in clinical trials, the inclusion and exclusion criteria, and how the conservatism that is maybe verging on stodginess scientifically could be standing in the way of increasing equity in clinical trials, which likewise has been a conversation for many years, but of escalating intensity, it seems, recently. So on that topic, can you tell us about the Leo cohort study presented at ASH and what it means?
Yeah, I actually, this is one of my favorite topics to talk about, Damien. And let me give a little bit of background also on why I'm particularly drawn to this. Years ago, I worked with a PhD student by the name of Abby Statler, who actually was working in the clinical trial compliance arm of my institution. And one day she came to me and she said, kind of shaking her head, and she said, there's something funny that's going on with these clinical trials. And actually, I write about this. Stat News was generous in hosting a podcast with me about the book I just had, Drugs in the FDA, that came out. And I described this in that book. She said, something's funny that's going on with these clinical trial eligibility criteria. And I said, what do you mean? She's like, well, they're exactly the same trial to trial, like word for word, exactly the same, regardless of the drug that's being tested. And I said, so you're saying it looks like they're a cut and paste job. Someone cut and pasted them from one protocol to another. And she said, yeah, that's exactly what it looks like. And I said to her, because that's exactly what they did. When someone's working within a company or within an institution to write a protocol, a protocol is a long document, right? And if it's an investigator-initiated trial, it's often 30 to 40 pages. If it's something that comes from a sponsor, a protocol can be 100 pages or even 150 pages long. So you put in sections, you beg, borrow, and steal a lot of stuff. And some of that I think is perfectly legit. If you're looking at trials in diffuse large B-cell non-Hodgkin lymphoma, I think it's fine to put down a background from one trial to another that says diffuse large B-cell non-Hodgkin lymphoma is a life-threatening disease. Or if you're looking at one specific drug from one protocol to the next, I think it's okay to put some of the same preclinical work that justifies the clinical work from one to the next. But what investigators will also do is cut and paste the eligibility criteria. And Abby asked me, do you think there's a way that we can show that it's a cut and paste job? So what she did is looked at about 100 randomized controlled trials in hematologic malignancies over a 10-year period, and looked at whether the eligibility criteria on these trials reflected the known toxicities of the drugs from the earlier phase trial or preclinical work, or the actual toxicities that were realized on those studies. And she actually showed statistically they had nothing to do with it. So eligibility criteria were restrictive for patients who had kidney disease or liver disease or heart disease, and none of the preclinical trials or preclinical work or early phase trials actually showed that there were kidney, liver, or cardiac toxicities to the drug. Yet they were excluding patients from these studies. Abby then took this to the next step, and we looked at 10 years of clinical trials out of the Southwest Oncology Group in leukemia. And as it happens, after the fact, some patients on those studies were found to have been ineligible. They shouldn't have been enrolled in the trial at all, yet they were enrolled because it was only after the fact that this was discovered. And it was actually about 10% of patients enrolled on those trials. So she looked at whether those supposedly ineligible patients who were treated on the trials actually did worse than those who were eligible and treated on the trials. And it turns out that across all of these trials in leukemia through an NCI sponsored cooperative group, patients didn't have increased rates of adverse events, of serious adverse events, and they didn't have changed rates in remission rates. So it turns out that these so-called ineligible patients could have been enrolled on the trials to start with. And as I mentioned, it was 10% of patients enrolled on these trials. Who knows how many didn't even make it to that stage, were excluded right off the bat, but could have been included. So take all of that background information about this, and what the Leo investigators did. This is first author is Khurana, but this is a consortium of lymphoma epidemiology outcomes that's conducted over, I think it's over 8 institutions, including our own, where these institutions combine their data to try to answer these sorts of questions. They looked at patients who were diagnosed with diffuse large B-cell lymphoma from 2015 to 2020 and identified 2,510 patients. They then looked at the eligibility criteria for different randomized trials in lymphoma, including the POLARIX trial, and applied those eligibility criteria to their cohort to see, gee, would they have been excluded from these studies, and what percentage would have been excluded from these studies. And what they found was really interesting, that all comers, there was a range of patients who would have been excluded from these trials that went from about 10% to 26%, right? 1/4 of patients who would have been considered for a trial wouldn't have made it on based on the eligibility criteria of studies that have actually been published. They then went on to make the distinction between non-Hispanic whites and non-whites. And I'm sure it'll come as no surprise to you and others that the rates of excluded non-whites was much higher, significantly higher than it was for whites. So in some studies, they came out, kind of similar, but in a couple of studies, there was a difference of, 12 percentage points or higher, and up to 35% of non-whites would have been excluded from those studies. So the conclusions from the authors, and this is something that Dr. Statler and I actually discussed in our discussion sections of the studies that we published. One was published in leukemia, the other one in blood, was that the eligibility criteria themselves probably, not only do they prevent certain patients from enrolling on those trials, but they probably, at a disproportionate level, exclude patients who represent non-white groups.
Hmm. And we got a question from William Wyndham sort of on that topic. I mean, his specific question was, how would you tell the difference between progression of a prior heart disease and a safety signal in a study? But I think, kind of extrapolating that, digging into that study, a lot of it is comorbidities that maybe didn't need to be exclusion criteria. But I guess the question is kind of in a clinical trial where, conservatism is not always ideal, but you want to isolate as many variables as possible. How do you kind of thread the needle where you are ideally maximizing inclusivity and equity as it relates to identity, as you just described, but also not compromising the final data of your study? Because what you really want to know is whether this intervention actually works.
Yeah, it's a great question, and I'm really glad that we were asked that question. It's one that I actually posed in my book, and one that I actually used that specific example of somebody with heart disease and how you can tell that. I think you start with a drug's known toxicities or highly suspected toxicities. And if that doesn't include things like cardiac effects or liver effects, you really should broaden your inclusion criteria to allow those patients onto it. Part of that also gets with whether or not an adverse event, particularly a serious adverse event, is likely to have been attributed to a drug or not attributed to a drug. So I think that we can have a little more oversight about that. If you think about it, Damien, when I participate in clinical trials and a patient has an adverse event, it's really up to me as the site investigator to determine attribution. And I'll tell you that sometimes I'm questioned about that by sponsors of trials, but sometimes I'm not. So I think that's also part of it. And I think we probably need a little bit more guidance to investigators on how to think through attribution to drugs, because we really have almost no guidance on this. This is particularly challenging for trials that give multiple drugs to patients. So when you're talking about some of these new myeloma trials that have 4 drugs, how are you going to describe attribution of one drug versus another drug if that same patient has heart disease? My final response to that is, particularly when I was practicing in Cleveland, it was the rare patient who didn't have heart disease. So when we're using these drugs in real world settings, I think we're setting ourselves up for failure if we're not enrolling patients with comorbidities onto these trials. And I would be in favor, for example, of like a no fault third arm that's allowed onto studies, right? And this is something that, I think FDA has kind of toyed around with, and they talk a lot about real world data and enrolling real world patients to studies, but they haven't gone all the way to say, and we won't hold it against sponsors if there are more adverse events in those cohorts, right? And it's kind of a risk benefit analysis. I think there's a way of actually doing this. I think you can say in certain patient groups, this is the risk benefit analysis. In other patient groups, this is the risk benefit analysis, and you should think about the drug differently.
Yeah. So speaking of the FDA, you wrote a whole book, as you mentioned, called Drugs and the FDA, available at all fine booksellers, I imagine, which is about the evolution of the agency and, many aspects of how, thalidomide to the present. But specifically, you have a lot of experience with the oft-debated accelerated approval pathway, which is that by which drugs can reach the market based on preliminary evidence or a surrogate endpoint that would likely predict benefit, but that will be confirmed at a later date in studies to come. I know that's a can of worms, but I mean, kind of focusing even just on like heme-onc, we've seen in the past year or so some negative confirmatory studies leading to product withdrawals in oncology in general. And so kind of getting to ASH and some of the therapies that will be discussed in part 2, a lot of the really promising data we've seen from investigational medicines are from these single-arm, relatively small, relatively short studies that will be submitted to the FDA and based on precedent will likely lead to accelerated approvals. And so I guess, kind of taking all that holistically as a clinician who is also steeped in sort of the regulatory aspect of this, how do you approach potentially prescribing these medicines once they clear accelerated approval, knowing all of the caveats that comes with?
Yeah, it's a fabulous question. So at the American Society of Hematology annual meeting, we see this like incredible science, right? And we see these amazing response rates, and we all get so jazzed when we leave the meeting to think about how we're going to apply this to our patients. In my field, my specialty is acute myeloid leukemia and myelodysplastic syndrome, so I'm much more focused on myeloid malignancies. What we saw was a large number of trials focused on patients with AML who are quote unquote unfit for intensive chemotherapy. These tend to be older adults, and we're seeing the emergence of a lot of triplet combinations. So with an azacitidine venetoclax backbone, and then an additional drug on top of that. And does the addition of that additional drug make aza plus ven even better? And we saw a lot of data that came out looking at overall response rates, and I think that is becoming increasingly complicated in acute myeloid leukemia. And the reason is that, first of all, even a CR rate, let's take the best response you can find, a complete response in acute myeloid leukemia. That's wonderful. That's what we're all shooting for. We think that that's associated with eventually leading to an improved overall survival. But there have been a number of studies conducted in older adults with AML that actually show that increased CR rate doesn't always translate to an improved overall survival. So one of my bugaboos as I'm hearing presentations at ASH is to question the investigators on duration of response or progression-free survival or something that's a little more meaningful of an interim marker that you think may translate to eventually to an improved overall survival. The other aspect of it in acute myeloid leukemia is we're increasingly seeing the use of terms like morphologic leukemia-free state that's added to overall response rate, or in myelodysplastic syndromes, a marrow CR. Both of those are basically the same things. It means you gave a drug, it reduced the blasts in the bone marrow from some higher number. If it's MDS, let's say 17%. If it's AML, it might be 40% to less than 5% without recovery of counts, with nothing else happening. So we showed in one study a few years ago that a marrow CR in MDS actually is no different than stable disease. It shouldn't be a response at all. So I really think, Damien, that one of the things that we can do, I think, as a community, and this includes, frankly, investors, is to discount things like morphologic leukemia-free state or marrow CRs from those overall response rates, because I think they're artificially inflating our perception of whether a drug is reasonably likely to translate to an improved overall survival. If we de-emphasize those at earlier phase trials, then I think those earlier phase trials are less likely to go to more advanced phase trials and then be at this step of regulatory approval based on accelerated approval, where eventually they're finally going to have the adequately designed randomized trial with an overall survival benefit that may flop, right? I mean, we're wasting years of effort to get to that point and billions of dollars of investment to do that. So I think as a community, we can kind of, at a very easy level, I think, start to discount some of those so-called responses that really aren't responses.
That's fascinating. I mean, I wonder what would be, I realize I'm derailing what I was talking about before, but what are the levers by which, like, how does the community come together there? Because you have obviously the FDA, which is making the final decisions, and they engage with stakeholders and advisors and et cetera, but you have sponsors who have many interests, among them making a product that will get to market as quickly as possible and get a return on investment. And then obviously patients and patient groups for whom the rapid availability of potentially transformative medicines is truly a life or death issue. And so kind of swirling all around that, how does something like this get broached? Where do you find the middle ground that theoretically, not necessarily pleases all parties, but theoretically has like the greatest like utilitarian good of speeding the development of medicines that are good without wasting money on medicines that turn out to be useless?
Yeah, it's complicated. So I don't want to come across as saying I have the answer, and if we just did this, it's everything, right? It would be wonderful if that were the case, but I'm not naive to the fact of how complicated this is and how many people have a vested interest in this. I'll give you a brief anecdote. When I was interviewing to join ODAC, so I was on the Oncologic Drugs Advisory Committee for 5 years, and I chaired it for 2 years. I sat with Rick Pazdur, and I once said to him, who of course heads up the cancer branch of the FDA, do you ever feel bad about rejecting a drug when you know it means a company will go out of business? Right? A valid question, but maybe one that he's not asked all the time. And he shook his head and he kind of smiled the way Rick does, right? Because he's so knowledgeable about all the stuff and he's done it for so long. And he said, advisors are probably some of the smartest people who understand the science and the true activity of a drug, financial advisors. And he said, if a company has one drug that doesn't work very well, financial advisors will know it. That's the reason they go out of business, that they're not getting invested in because the drug doesn't work that well. He said drugs that work really well don't have a problem. So I've always thought about that, right? Because there is kind of a role, I think, of investments in these companies for drugs that don't work very well. And that's why I kind of pitch this as saying, well, us in the scientific community, I think we've got to hold ourselves accountable for responses that really aren't great responses. But I also think there's some responsibility from the business community.
That's interesting. That's fascinating. That everyone can conceivably benefit from a drug that actually works, because at least the way the system is ideally set up, the inventors of that drug will be monetarily rewarded in time if they do the job right.
Right. Exactly.
Awesome. Well, that is actually, we're just out of time for this part one of this, and it is a good segue to part two. So first of all, I'd like to thank you, Dr. Sekaris, for taking the time to chat with us, and I will pass it over to my colleague, Angus Chen, for part two of our ASH recap.
Thanks so much, Damien. Bye, everyone.
Thanks, Damien. And so here to join me for the second portion of today's event, please welcome Dr. Laura Michaels. She's Professor of Medicine at the Medical College of Wisconsin and Chief of the Division of Hematology Oncology at Froedtert Hospital and Editor-in-Chief, The Hematologist. I'm hoping I said that all right, Dr. Michaels.
Thank you so much, Angus. Yeah, actually, I just finished my editorship, and The Hematologist is now being led by Dr. Shashi Kumar, an expert in myeloma. So I'm a myeloid person, and it's moved over to a myeloma person, but hopefully people will continue to get great information from that publication.
Yeah. So, for this session, we're doing, I guess, a little bit of a spotlight on leukemia, since there were a number of exciting and I think in some cases practice-changing abstracts at ASH this year on leukemia. But one of the things that I wanted to start with was the presentation of a discovery of a monoclonal antibody with the very long name INSA 033989, but it looks like it might have some use for myeloproliferative neoplasms, type of blood cancer that starts in the bone marrow, right?
Sorry, go ahead.
So I guess just what made this abstract important to you, and what's its significance to MPNs?
Oh, yeah, I'm so glad you brought this up, because this was one of the plenary abstracts, and because I treat myeloproliferative neoplasms, I was particularly interested in hearing this data. So MPNs are a relatively rare form of leukemia. They can be either, in rare cases, very aggressive. Typically, they're really chronic, but that doesn't mean they don't have a lot of effect on people. People can get thrombosis of both the arteries and the veins. They can have a lot of symptoms. And it's been a disease that is definitely or historically been managed with symptom management. Now, there's about, as I say, about 300,000 people in the United States with MPNs, and it was in 2005 that one of the mutations that takes somebody from having a normal bone marrow to a bone marrow populated by these overproducing cells, the JAK2 V617F mutation, was described in 2005. And I'm sure the people listening to this are aware that it was only 6 years before the first treatment for MPNs in the form of a JAK inhibitor came out. Now, one of the really interesting things about JAK inhibitors is that the way that they've been approved and the efficacy that people see in patients is a reduction in symptoms and a reduction in the spleen volume. So the spleen, as kind of an annex for your bone marrow, is where a lot of bone marrow production happens in myeloproliferative neoplasms, because the bone marrow itself gets so crowded, it has to find other space to make blood cells. And that really speaks to the fundamental problem in MPNs, is that blood production gets decoupled from the signals from the rest of the body that say you need to have blood production. And at the cellular level, at the JAK2 mutation, that happens inside the cell. But in 2013, we learned that there's also another way that the mutations can happen in the stem cell, and that's with something called mutant calreticulin. So calreticulin is kind of an interesting protein. It typically sits inside the cell, and it means that the promoter, the part of the cell that allows the part of the stem cell that allows it to receive signals called thrombopoietin, it keeps that cell that receptor inside the cell until needed. So it kind of chaperones it inside the cell and then normally lets it come out and be seen on the outside of the cell as a place to receive signals that says, oh, this person needs more platelets made, for example. Well, in patients with essential thrombocythemia and myelofibrosis, about 30% of those patients, that CALR is mutated. So instead of working well inside the cell to keep that thrombopoietin captive inside the cell, it lets it go, and it lets those cells now have tons and tons of antenna out there that say, wait, I think we should be making platelets. I think we should be making platelets. And a result, the people that have myeloproliferative neoplasms with this mutant CALR have platelet counts, instead of in the 200s, in the millions. And they have a higher rate of myelofibrotic transformation. they may or may not have an increased rate of acute leukemia associated with it. That's typically more seen with JAK2. But these people can have platelet counts that are exceedingly hard to control, leading to symptoms, leading to clots in their arteries, clots in their veins, leading to pregnancy loss, etc. Now, this study, which was presented by Dr. Admira Reese, who actually is a scientist, a basic scientist at Incyte, was on the activity of this drug that has been developed, a monoclonal antibody, just like we have monoclonal antibodies like rituximab or obinutuzumab. This is the first monoclonal antibody that's been looked at in myeloproliferative neoplasms. So it's really groundbreaking. Now, it hasn't yet been tested in humans, but what this antibody does is when that antenna goes outside of the cell, let go because of the mutant CALR, this antibody keeps the antenna from actually functioning by sitting in beside it. So it can't produce the signals that say make more platelets. And by binding that mutant CALR, and it doesn't bind any normal CALR elsewhere in the body, so the therapeutic window is really good. And what this plenary session showed, and the data is beautifully done, is that there's both in the cell line models and in the mouse models, there's a dose-dependent decrease in the intracellular machinery that is required to then go on and make platelets. So the more antibody that's on there, the less likely that those mutant cells are going to release things like STAT3, STAT5. These are the kind of signaling molecules that then overproduce platelets, that can cause scar tissue in the marrow, leading to fibrosis, that can cause the body to need to increase the spleen size, etc. So it's selective. It works. It appears to work based on the data that was shown. It appears to work in cell lines in this dose-dependent manner. It appears to be very specific for the mutant CALR, leaving the normal CALR alone. And it also decreases and changes the balance of mutated stem cells versus good stem cells in the marrow. So we know in people with MPNs, the percentage of mutated stem cells goes up over time and the percentage of normal stem cells goes down. So that's how those diseases progress. But it appeared, based on some of the studies, shows that this selectively decreases the mutated population, so you can rebalance the bone marrow, which is, if that's the kind of outcome that we can measure in a clinical trial, which is, it hasn't yet moved to human trials, the phase 1 is planned for 2023, but if we can show that, then we can really look at changing the disease itself as opposed to just treating symptoms, which is something that's kind of the holy grail for MPN patients. Instead of just putting a Band-Aid on it, can we safely and selectively change the way those marrow work and revert people to normal disease, which would be an incredible benefit for patients if this kind of technology, this kind of idea pans out. So I think that's what was really exciting.
Yeah, I mean, I think it's certainly the ability to really target this cancer, but not just that, but like cancer that really doesn't, like you said, have a lot of treatment options.
Exactly.
But so there's a bunch of abstracts that we want to get to. So I'll move on to the next one, which is not just about not about new drugs or potential new drugs, but kind of old ubiquitous drugs. This year, I think there was one trial that showed that ibrutinib, which you and others described as the king on the block when it comes to frontline treatment of cancers like CLL or chronic lymphocytic leukemia, might be getting dethroned, perhaps to the disappointment of AbbVie, which makes this drug. So what did you think of this work, and could you give us a little info around it?
Yeah, so the Alpine study, which was presented by Dr. Brown in the late-breaking abstracts, was one of those other really great, I think, very well done studies. So first off, I'm not taking anything away from ibrutinib. Ibrutinib has really changed the way CLL is treated. It has provided so many great treatment options for people who previously had to be treated with cytotoxic therapies with a later risk of secondary leukemias, for example, and with high death rates and low overall progression-free survival. So ibrutinib deserves all the kudos in the world for changing the way that CLL is treated. And like all these novel agents, there's always sort of the second generation. And that's what Alpine was looking at. So Alpine was looking at the use of zanubrutinib, which is a second generation butyrosin tyrosine kinase inhibitor with some higher specificity than ibrutinib, with a change in the way that the blood metabolizes it. So the PK doses, you basically stay at a therapeutic level for a little bit longer period of time when compared to ibrutinib. So zanubrutinib had been compared to chemoimmunotherapy in the past, but this is the first time that it really was looked at head to head against ibrutinib, which we know is the first generation covalent BTK inhibitor. Now, ibrutinib has some challenges in administration, and that is reflected in the number of people that have to come off of treatment. And sometimes they come off of treatment because of bleeding, which is really a class effect that I'm not sure that we've gotten away from. But you can also come off of it because of cardiac events. And the rates of atrial fibrillation, especially in older people who are the ones who have CLL, has also been noticed in this. So this study wanted to ask the question in people with relapsed or refractory CLL, SLL, who had not been treated with a BTK inhibitor, meaning that maybe they got venetoclax, but most commonly they got chemoimmunotherapy. Do patients do better, or is there a benefit to using zanubrutinib rather than ibrutinib? So Dr. Brown presented about 650 patients with a overall, I think about almost a 3-year median follow-up. And there were 327 on the zanubrutinib arm and 325 on the ibrutinib arm. And what she showed is that there was a significantly favorable 2-year progression-free survival in favor of the zanubrutinib over the ibrutinib arm, with the 24-month cutoff of progression-free survival for zanubrutinib being 79% and for ibrutinib being 67%. And that was also true in the 17p or p53 mutated disease, which we know is sort of a bad player in this, and also, like it is in many of these conditions, more likely to relapse. And then importantly, I think in a disease where you're not necessarily looking for complete remissions all the time, and you may not be looking for cure, what you're looking for is disease control in a safe manner. There was also an improvement in serious adverse events, especially cardiac adverse events, and improvement in the rates of atrial fibrillation. So for example, the zanubrutinib arm had a 5.2 risk of atrial fibrillation, and the ibrutinib arm had a 13% risk of atrial fibrillation. And when you're thinking about, well, why does that matter? Atrial fibrillation is not life-ending, et cetera, but it is actually a big deal in patients with CLL on these therapies, because once somebody gets atrial fibrillation, they often have to be anticoagulated. And we know that one of the class effects of Bruton tyrosine kinase is that the platelet clotting time is prolonged and people can have bleeding. That's why the exclusion criteria for people on warfarin. But now you have somebody who's on a drug that now they need to be anticoagulated. So now you're actually increasing the bleeding risk associated with it.
And
And
like a double whammy on
exactly.
on the bleeding.
Well,
yeah.
and it leads to like a cascade of additional therapies that people have to be on. Oh, now I have to be on a rate controller. It's like a heart rate controller. Now, hypertension did occur. It didn't seem to be different between the two arms. As I say, there were more serious adverse events related to cardiac problems, sometimes atrial fibrillation or other rhythm abnormalities. So, and the other major grade 5 effect that happened on both arms was death from COVID, because this trial was run during the pandemic, and we all of us who treat CLL know how devastating the pandemic was for these patients who are really immune suppressed. So this has been published in the New England Journal. It was sort of published concomitantly. I haven't read the full publication yet, but I do think that this, again, gives a lot of reasons to think about whether or not you want to consider a second generation rather than a first generation BTK inhibitor in your patients. I think this will be the kind of thing that's debated at our kind of planning committees at hospitals around, what's our next step for these patients.
Yeah, I mean, when I talked to Jennifer Brown about this, she was like, you know, she couldn't think of a patient population in which you would choose ibrutinib over zanubrutinib or even acalabrutinib, which is another second generation BTKI.
Right.
I don't know. Do you feel like really the studies are showing that the first generation BTKIs are kind of obsolete now?
Well, I don't want to go so far as to say obsolete.
Mhm.
And Jennifer certainly has a ton more experience in CLL than I do, so I will defer to her for the expertise. But I do think when I'm talking to my patients, I'm going to need to take into account very, very significantly some of the benefits of these second generations, in particular with regard to cardiac events.
Yeah. And just I don't know if we mentioned this. Ibrutinib made by AbbVie, zanubrutinib is made by BeiGene. I wanted to move on sort of maybe further along in sort of the course of a leukemia patient's treatment to when they might be thinking about hematopoietic cell transplants or allo transplants. One of the abstracts that really caught my eye was this late-breaking abstract from Dr. Mark Litzow at the Mayo Clinic on acute lymphoblastic leukemia, who sort of looked at the addition of a bispecific antibody in ALL consolidation therapy. And it was interesting to me because it seems to suggest that some of these patients might be able to really avoid stem cell transplants with the addition of this immunotherapy.
Absolutely.
Yeah.
This was a great study. So this is one of the U.S. cooperative group studies. It's led by the ECOG Akron. And in addition, actually, there were participants from Israel and Canada on this. But this was a study, a long time coming. It was actually written in 2013, and then data cutoff was 2019. But this asked the fundamental question that was presented at late breaking abstract by Dr. Litzow is, in the MRD negative patients, so these are patients with B-cell ALL, and they get an induction regimen, a multi-agent chemotherapy induction regimen that we refer to as the 1910 backbone, but it's really a German backbone called the BFM backbone. Those patients, they get induction, so they hopefully get into a CR. They go through an intensification to make sure that they don't get CNS disease with high-dose methotrexate. And one of the key questions there is, are those patients MRD positive or MRD negative? Now, we know that Blina was approved by the FDA for the MRD positive group. So what we're looking at now are just the people that are MRD negative. And in those people, they were then randomized to have blinatumomab and intensive combination chemotherapy given during this next phase of treatment, or just combination chemotherapy followed by a maintenance thing. So both arms got combination chemotherapy. Both arms got maintenance. The duration of treatment was the same, meaning that the Blina arm got some of their maintenance taken off. But the Blina arm also got basically, I think it ended up being 6 weeks of blinatumomab, 4 up front, and then 2 additional weeks kind of staggered later on. And what Dr. Litzow presented was that of the 224 eventually who trickle down and are found to be MRD negative, 112 got Blina, 112 got chemotherapy, and that the median overall survival of those with Blina wasn't reached, and the relapse-free survival wasn't reached. There were only 17 deaths in that arm as opposed to 39 deaths in the chemotherapy alone arm. And so significant hazard ratio in favor of Blina. Now, do we know if those people don't need transplant? No, we do not, because that wasn't one of the questions. Physicians who put patients on this study needed to say upfront, this patient ought to be transplanted or this patient shouldn't be transplanted. And the ought to transplant patients could go over to transplant anyway. So we don't know if these people need to be transplanted, but we do know that compared to what we could traditionally tell folks, well, this is your likelihood of overall survival, median overall survival is in the 70-month for you. We expect that half people will relapse in 22 months. We can't say that anymore because now we have this way to keep people in negative much longer, which is really great data and very, very encouraging. Most of us, we use transplant all the time, but we don't want to have to.
Yeah, I mean, I think one of the physicians that I spoke to about this trial mentioned that transplant can have some pretty rough side effects for toxicities for patients. In some cases, even the risk of short-term mortality. So, yeah, the opportunity to maybe delay that or not have it at all with blinatumomab, which is made by Amgen, I think, is really significant.
Absolutely. So
Yeah.
avoiding transplant is a very good goal, especially in ALL. And I mean, I think when I think about where the field is going to, one of the main goals of the field of B-cell ALL is to be able to de-escalate the therapies that are the traditional cytotoxic therapies that have long-term risks for MDS or leukemia treatment-related myeloid neoplasms, for example. And so moving towards targeted agents, moving towards immunotherapy like blinatumomab, which links CD19 to CD3 so that asking the T cells to kill those CD19 cells. That's one direction. And the second is to avoid transplant. Remember, B-cell ALL is most common cancer in children. So the more we can learn that can safely eradicate the disease without giving people risks for long-term health impairment from those therapies is incredibly important.
I want to make sure that we do answer a question from the audience. There's
Sure.
one from Angela Madelon about ibrutinib and zanubrutinib. She wants to know if it would be helpful to use a non-covalent plus a covalent BTKI up front, so I guess ibrutinib or zanubrutinib, to delay resistance.
Yeah, so great question. That hasn't been tried. Right now, BTKs are being used just as a single agent. I think what we're going to see and we already have seen in combinations is whether or not we get more bang for our buck and in terms of reduction of measurable residual disease in CLL by combining BTK inhibitors with BCL2 inhibitors. So you know that there's triplet therapies right now that include a B-cell antibody like obinutuzumab or ofatumumab, for example, with a BTK inhibitor, ibrutinib, and with a BCL2 inhibitor like venetoclax. And probably more to come with those multiple combinations. I don't think we know yet in CLL, though, whether or not the better thing for the patient is to hit them hard early to get the disease as low as possible to get to MRD negativity, or if our goal should be to use less therapies, one after another, after another, after another, to basically hold the disease at bay, hold the consequences of it at bay, but not necessarily expose people to the depth of treatment and the risk of problems with it. So the question of like low and slow or fast and hard has not yet been answered in CLL. And I think that's one of the things that our community needs to address.
With that, I think we're just about out of time. Dr. Michaels, thanks so much for joining us and taking the time to speak with us about these exciting abstracts at ASH. And I want to also thank Dr. Sekaris for joining us today and answering some really great questions from Damien and our audience. And thanks to our event sponsor, Janssen Oncology, and to all of you out there for taking the time to join us. Be sure to join in the new year for our Stat at JPM live event taking place in San Francisco on the evening of January 9th. So more information about the program and how to register can be found on our website. Thank you again. Stay safe and be well.